From this video, we are starting with the new unit, that is genetics and evolution. In this unit, there are three chapters. The first one is principles of inheritance and variations, where we'll be discussing about Mendel's principles and various kinds of crosses. The second chapter is molecular basis of inheritance, where we would be discussing about the main molecule that is, or genetic material, that is DNA. Then various processes of transcription and translation. And the last chapter is about evolution. And here we'll be talking about various theories of evolution. So let us start with the first, that is the genetics principles of inheritance and variation. When we talk of genetics, the first thing that comes to our mind is the father of genetics, that is Mendel. Father of genetics is Gregor Johann Mendel. Mendel was born on 22nd of July in 1822. That is the birth year. In 1822, for 6 years he worked on Pisum sativum, and that is the work which has given us all the principles of inheritance. So 6 years of work on Pisum sativum, that is the pea plant on which his work was based. He died, that is death year was 1884, and till he died his work was not recognized. We'll discuss about the reasons why his work went unrecognized for such a long period of time. 16 years after his death, 16 years after his death, 3 scientists rediscovered Mendel's work, and that is when the work got recognition. So 16 years after the death of Mendel, that is in 1900, 3 scientists, their names were Hugo, DeVries, Correns, and Tschermak. These 3 scientists, they rediscovered Mendel's work, and that is when he got this recognition for his work on the pea plant or Pisum sativum. Mendel was not a biologist. He was a mathematician, and he was assigned a job of a priest in a church. So the reasons why Mendel's work went unrecognized for such a long period of time, there were 3 very important reasons for those, or for that. So let us take those reasons why his work was not recognized. The first reason was that during those days, the methods of publication were very simple and ordinary. So the research that he did was not published on a larger scale. So we are talking of reasons for non-recognition. The work was not published properly. Now, what do we mean by publication of work? There are local journals in which the research work can be published, and there are international journals in which the work should be published. Now, if we publish our research into a local paper or a local journal, it would reach only a limited number of people. To make it globally known, it has to be published in international journals, which was not done by Mendel. So that was one major reason. Second reason was because he was a mathematician, biologists were not able to comprehend this concept that a mathematician is explaining us the biological processes. So biologists did not accept the work properly. Did not accept. And at the same time, they were also busy discussing Darwin's work at that time, which was like known everywhere, and people were very much excited by Darwin's work. Third important reason, Mendel was talking about factors which we now know as genes. So these factors which Mendel was talking about, that these are responsible for inheritance of character from one generation to the next, these factors were not understood. These were discrete units. And people were not able to understand how these units are being passed on. Now what we mean by these discrete units is that genes, which we now know as genes, when Mendel said that factors are the things which are responsible for a particular character. At that time, microscopic work and all those things were at a very, very elementary level. So people had no idea how a DNA looks and how the genes would be. So to understand something imaginary, that there's something in the body or in the cell which is being passed on from one generation to the next generation. So this idea was not actually very convincing to people. So these three reasons were considered as the most important reasons for the failure or non-recognition of Mendel's work. Reasons for non-recognition. Now later on, Mendel's work got recognition, as we just now said, that three scientists in 1900 rediscovered, and that's when Mendel's work got recognition. Now there are reasons why his work was taken in a very nice manner, and why till date we are following the same principles. So now we are talking of reasons for Mendel's success. First reason, he selected contrasting characters. Selection of contrasting characters in pea plant. Now what exactly is meant by contrasting, and why was this a very, very important point? If we talk of a particular character, say height of the plant, and if we say one plant is tall and the other plant is taller. Now here individual perception is going to come in. For one person, one particular height of a plant could be tall and the other height of the plant could be taller. For other person, one could be short, other could be tall. So here there is a vagueness or ambiguity. In case of contrasting character, one plant is tall and other is dwarf or short. So short is short for everybody and tall is tall for everybody, or color for that matter. Suppose if you are talking of color, and if we say one is purple, the flower color is purple. One is dark purple and the other is light purple. Now here again individual perception is going to matter. But if the flower is purple and white, there can be no ambiguity or confusion. Purple is purple for everybody and white is going to be white for everybody. So that was the significance of contrasting characters, and he selected 7 such contrasting characters. We'll talk about all those 7. Another important reason for his success was a huge sample size or large sample size. That means every time he collected his information from various plants, the number of plants or number of case studies was very, very big. Whenever we want to give any kind of conclusive report, the number has to be huge. If we say amongst 10 people, the eye color was black and we say this was dominant, then this is not going to be acceptable. Unless and until you survey say 10,000 people or maybe 1,000,000 people and then come to a conclusion, then your result would be considered as more authentic. And so here this large sample size was a very important thing. Another important reason for his success was all the data that he had was analyzed in a very systematic manner using mathematical roots. As we said, he was a mathematician. So, nice or correct data analysis. And fourth, we can say it as a chance thing or he purposefully did it, we have no idea. But the reason why he succeeded in all his research work was that none of these characters were showing linked genes. So there was no reference or use of linkage. Probably if some gene was a linked gene, then the ratios or the conclusions which he drew probably would have changed. So there was no reference of linkage. He did not talk about any linked gene. The data analysis were all statistical, mathematical. Sample size was big and he selected contrasting characters, so there was no scope of confusion. Now we have discussed the reason for his work getting non-recognized for such a long period of time. After other scientists contributed, his work was recognized, appreciated, and these were main reasons for the work getting appreciated. Now let us talk about why Mendel chose, or why did he choose pea plant. So reason for choosing pea plant, as we were talking about, that he was working as a priest in a church, he was responsible for taking care of the complete premises of that church. And in the backyard, there was an open land where he was growing this pea plant. So, reasons for selecting pea plant, that is Pisum sativum. So the most important reason, the lifespan is short. Pea plant completes its life cycle in about 3 to 4 weeks. So in 3 to 4 weeks you are able to get the next generation, and if you are able to study many generations, again you are getting large sample size and your data is going to be authentic. 2. Pea plant shows self as well as cross-pollination or fertilization. Self and cross-fertilization is possible with pea plant. So probably that was one reason why he selected pea plant. Third, pea plant also shows contrasting characters. Contrasting characters. There are 7 contrasting characters which Mendel selected. So we will write down these contrasting characters. The character, we will write characters, and then we will also see which particular trait or which particular expression is a dominant one and recessive. So here we are talking of the character that we are talking, dominant, and which one is recessive. First one was height of the plant. Tall was dominant and dwarf or short was recessive, and contrasting. So tall and dwarf, there is no ambiguity. Second, the flower position. If the flower grew in the axil, then it was called axillary, and axillary position of the flower was a dominant character or is a dominant character, and terminal position of the flower is a recessive character. The third character is flower color. the color of the flower. Purple is dominant and white is recessive. Fourth, the pod shape of the pod. Inflated is dominant and constricted is recessive. Next is pod color. That is, green was dominant and yellow was recessive. Then sixth character, seed shape. Round was dominant and wrinkle is the recessive one. And the last character, the seventh one, that is seed color. Yellow is dominant and green is recessive. So these were the 7 characteristics which Mendel chose, and these characteristics showed total contrasting characters, and here there can be no confusion. Inflated is the pod which is going to look like, suppose we are talking of P, the pod is going to look like this, the inflated pod. Whereas in case of constricted, it will show these kind of constrictions. So there can be no confusion. Color, one is green, other is yellow, so there is no ambiguity or vagueness. So the reasons why Mendel selected P were some important were that short lifespan, it could be fertilized using the pollen grain of the same plant or of another flower, and distinct contrasting characters. Plus, growing pea plant is very easy, doesn't require that much of maintenance. One more important point about pea is that the seeds, let us write the 4th point here, the seeds do not show dormancy. Do not show dormancy. That means as soon as the seeds are formed, they germinate. We know what dormancy is. It is the period when, in spite of giving all the favorable conditions, the seeds will still not germinate. And if that is the situation, we have to wait for that period to get over, or we have to break the dormancy. But that is not the situation with pea plant, and that is also an added advantage. One more reason is pea plants, they produce large number of seeds, so the next generation which we get gives us a more or a bigger sample size. So number of seeds produced per generation or per plant is more, and this helps us get a bigger sample size. So these were the reasons, and these are the characters which Mendel chose, contrasting characters. In the exams or whenever we take a test or any examination, we are not told that this character is dominant or this character is recessive. We are supposed to remember at least these 7 characters. So this simple table is very, very important for us to understand, because moving forward we will simply take that purple color flower was or plant was crossed with a white colored flower plant. So we know that purple is dominant and white is recessive. So we have seen the reasons why Mendel's work went unrecognized for such a long period of time. We also saw that why his work is so authentic nowadays after scientists rediscovered it, and the reasons also to select the pea plant. He selected Pisum sativum, and these were the 7 contrasting characters which he used. Now let us talk about certain important terms which we used when we discussed genetics.